Photosynthesis involves using sunlight to combine carbon dioxide and water into carbohydrate, that is, sugar. Light is captured in the leaf, which is bounded by an upper and lower leaf cuticle. Photosynthetic cells contained within the leaf capture the light energy in a tissue known as the spongy mesophyll. Plants get their water from the roots, drawing it up from soil water. Some plants can absorb water directly through the leaves, but this is generally a minor source compared to soil water. Terrestrial plants get their carbon dioxide from the atmosphere. To do so, the air spaces of the spongy mesophyll must be open to the atmosphere. This occurs through tiny pores in the leaf called stomata. Each stomatum is bordered by two guard cells, which can swell or shrink and either close off or open up the stomata. This causes a problem for desert plants. To capture carbon dioxide, the stomata must be open. Open stomata also means high evaporation from the spongy mesophyll to the dry atmosphere. In an environment where water is scarce, this can be problematic, to say the least. Desert plants have many ways to reconcile this balancing act. Some involve different styles of photosynthesis. One interesting mechanism involves sinking the stomata into pits on the leaf surface. This allows the stomata to allow carbon dioxide in while limiting evaporation. This seems paradoxical. Both carbon dioxide and water vapor are gases. How does sinking the stomatum into a pit limit the diffusion of one and not the other? The answer turns on the concept of the so-called limiting resistance. Let's break that down. Here's a simplified schematic of a spongy mesophyll cell, the upper leaf cuticle, and a stomatum. For a molecule of water vapor to move from the cell surface to the atmosphere, it must traverse the airspace of the spongy mesophyll, then through the stomata, then through a still air layer at the leaf surface called a boundary layer, and then to the atmosphere. How rapidly the water molecule runs this gauntlet, m.H2O, will depend upon the quotient of the magnitude of the water vapor partial pressure difference, delta pH2O, and a resistance to diffusion of water vapor. In the case of the leaf, the water vapor partial pressure differences will go from the wet cell surface, P sub H2O CS, to the spongy mesophyll air, P sub H2O SM, then to the leaf surface, P sub H2O-LS, and finally to the atmosphere, P sub H2O ATM. That resistance can, in turn, be broken down to the sum of a series of subsidiary resistances that correspond to the distribution of water vapor partial pressure. From the cell surface to the spongy mesophyll, R sub SM, through the stomata, R sub ST, and then finally through the boundary layer, R sub BL. The total resistance to water vapor loss will be the sum of these three resistances. The idea of the limiting resistance comes when one of these subsidiary resistances, let's say the stomatal resistance, is much greater than the other two. Then the total resistance to water vapor diffusion will be approximately that of the stomatal resistance alone. Stomatal resistance is therefore the limiting resistance. Variation in this resistance, as might occur by adjusting the stomatal aperture, will have a disproportionate effect on the leaf's water loss rate. This is the rationale between desert plants sinking their stomata into pits. This adds a fourth resistance to the series, the resistance between the stomatal opening and the leaf surface, or R sub pit. These pits usually trap still air, and this makes the resistance to water vapor diffusion across them large. This adds to the already large stomatal resistance, with the consequence that water loss rates through evaporation are reduced. How about carbon dioxide? Carbon dioxide also diffuses through the leaf, but in reverse. From the atmosphere, through the pits, through stomata, and into the mesophyll cells. As is the case with water vapor, diffusion rate is driven by gradients in carbon dioxide partial pressure. From atmosphere, P sub CO2 ATM, to the leaf surface, P sub CO2 LS, through the pit to the stomatal opening, P sub CO2 SO, into the spongy mesophyll, P sub CO2 SM, to the cell surface, P sub CO2 CS, all limited by resistances to diffusion. So far, this is looking a lot like the resistance network for water vapor loss, but there is an additional carbon dioxide flux from the cell surface into the cell itself, where photosynthesis takes place. In the cell interior, partial pressure of carbon dioxide, P sub CO2 CI, is low, reflecting the cells drawing carbon dioxide into photosynthesis. There will also be a resistance to the flux of carbon dioxide across the cell membrane, R sub cell. Total resistance to carbon dioxide flux will now be the sum of 5 terms, 4 of them the same ones that limit water vapor loss, but a 5th corresponding to the resistance to carbon dioxide flux into the cell. This latter resistance is the largest term in this equation, so it is this resistance that is the limiting one. This means that all the other resistances, the boundary layer, the pit, the stomata, and the spongy mesophyll, contribute little to the total resistance to CO2 flux. In this way, sinking stomata into pits will limit water vapor loss, but will have little to no effect on CO2 uptake. It's all a matter of which resistance is the limiting one. In the case of water vapor loss, the limiting resistance is at the pit and stomata. In the case of carbon dioxide flux, the limiting resistance is at the cell membrane.